So thanks for coming back to the second section, which is about being fair. So this is regarding the application to data. So what is data? Data is not only information that is recorded in an Excel sheet or in a structured data sheet, spreadsheet. Data is often also images, and also not only from microscopes. It can be information about biological materials or strains, or even patient details if it's more sensitive data. It can also be laboratory measurements, protocols, models that you use. Also, some scripts, analysis procedures, and custom software is also considered as a subset of data. So we want to start this with an exercise, actually. So we outlined the instruction to the exercise in the pad. If you can move to the bottom of the pad. So the exercise is that you need to imagine you're in a scenario where you have to do a Western blot to identify titin proteins, the largest proteins in the body, and it has a molecular weight of 3,800 kilodaltons. You find an antibody that's sold by Sigma Outreach that has been validated in Western blots and in immunofluorescences. Sigma Outreach lists UAL's 2019 paper for reference. So you can see here a paper link. So you can click on the paper, and the exercise, is that you need to find the protocol for doing a Western blot in this paper. And you now need to find details on how to separate and transfer this large protein in the paper. So there's some hints for you, which is to go to the method section, and there is a Western blot analysis subsection. You can follow that reference. And our question for you is, did you find that this method in this paper was findable, accessible, or reusable? Show you my journey. So first off, I found reference 17 in the first paper, the UAL 2019 paper, which led me to this one. And I look at Western blot here, and it says SDS polyacrylamide gel electrophoresis and Western blotting were performed according to standard methods. So I think that doesn't tell me much about really how it was done with this antibody that I'm supposed to use. So it really is a dead end, and it doesn't really help me with my experiments. So I think in this case, the only way would be to contact the authors and see if they have any type of records of this. Yeah, so that's the first exercise. Any reactions? Would you call this fair? Paper of paywall, and then in addition, there was no details. Yeah, so we have the next exercise. So I will paste this again into our pad. Okay, so exercise number 2. So this is a paper from ECRM 2014. And this paper contains data about various metabolites in different accessions. So you have several genotypes of an Arabidopsis plant. You would like to calculate the average nitrogen content in the plants grown under normal and nitrogen limited conditions. So how do you calculate the average over these genotypes and the nitrogen content in the 2 experimental conditions? So you have 2 hints. One is that the data is in the supplementary data. And 2 is search for nitrogen in the paper text to find the correct data column. A problem with having data in supplementary. So it's a lot less discoverable, a lot less accessible if the paper itself is not open access. So ideally, your data lives in a separate open database in addition to the paper, and not in the supplementary, not only in the supplementary files. The file I found doesn't even have labeling of what the columns mean. Right. To understand what those columns really mean, unless there's even a figure caption for the table or table caption. Yeah, so so this is our exercise 2, just to demonstrate if the data is not placed in a nice format or clearly labeled, it's impossible to reuse this research data, even though it's published. Okay, great. So the last one, I promise, the last exercise in frustration is looking for a link. So please see exercise 3 in the pad. So the challenge here is that so there is an RNA-seq transcriptomics data that is usually deposited on online repositories, such as SRA or Array Express. Your task is to find the link to the repository of the raw RNA-seq data in Lee et al. Gene Devs 12. So let me also insert the link. Sorry, 1 second. I'll insert the link to the paper below. Yeah. So let's all try to find the link to the repository of the raw RNA-seq data. There is something, but it was definitely not the easiest to find. Okay, so that concludes our exercises. So going back to our course material. Okay, so of course you saw this part. It was difficult to find the right data table and column, and the numerical data in a PDF is just not workable for any calculations. And yeah, so some common problems you can get is only average data is available, or you don't have any numerical data in a proper format. You can live in a PDF file with no supporting information. Yeah, so sometimes it links to non-existing links. Yeah, and the most common one that you can see is often data code on request. So yeah, I think you have all experienced this, not only in today's course, but also in the past, that the protocol to something was difficult to find and is difficult or sometimes even impossible to access legally. Or it's not reusable and it doesn't have enough necessary details. So yeah, the second example is a clearly wrong format, not interoperable. And the data was not clearly described, making it not reusable. So now you see that through these various frustrating examples, what is important to make it easier to find. And it should be accessible, so not living in a closed access paper. And to make it interoperable is to make it in a clear and reusable format, and also describe what actually the data contains, and that allows something to be actually reusable. So these are the 4 underlying elements of the FAIR principle. So without going into too much detail again, so what it helps make something findable is to have the data and metadata for humans and computers to be able to read, and that this should be automatic and reliable discovery based on certain services and machine readable persistent identifiers. So in your publication, if you include a persistent identifier to the data and metadata. And the second element is accessible, is that you can then, with this persistent identifier, using a standard and open communications protocol, such as authentication authorization, you can add that and allow the metadata to be available even when the data is no longer available. So the next aspect of interoperability is to allow the reuse of your data with other data and tools. So the format of the data should be open and interpretable for various tools, and applies for both data and metadata. So then the metadata can also follow specific vocabularies of the FAIR principles, and this we'll cover later in the course. And lastly, reusability is that FAIR aims to allow the data reusability to be optimized. So if the metadata and data are well described, then this analysis can be replicated. And yeah, so this is a general overview of the FAIR principles. And so this was already mentioned in earlier in discussion, that you can add your data to a database. So there's many repositories that allow and provide persistent identifiers to the data that you publish. They also have cataloging options. You can also search by metadata, and you can also obtain download statistics. So they can also host private data in addition to open data and provide embargo periods. And like mentioned earlier, if you have this data living in an external open database, then this can be more findable and accessible than if we're living in the supplementary file of a paper. So some of these data agnostic repositories are Dryad, Zenodo, Figshare, and Dataverse. Here you can obtain a DOI for your work, but sometimes these databases don't have good data or metadata standards. So ideally for your own data, depending on what it is, you should find your community supported, trusted database that's suited for a specific type of data and that have certain metadata standards. So we also have our own data resources. So if you go to this link, ebi.ac.uk/services, you can find a massive list of various types of data and which repositories it's suggested to use. So some of them are certainly domain specific and specific to types. So for example, protein data goes into UniProt or GenBank for genomics data. Okay, so another word about persistent identifiers is that the persistent identifier is a long-lasting reference to a digital resource. It often has two components. One is a service where you locate the resource over time, even though the location changes. And another is that it's unique. It distinguishes this specific data from any other data source. So one example of that for paper is the DOI, digital object identifier, that is specific for a paper. There are several services that provide persistent identifiers like digital object identifiers. And so this, for example, will lead you to the FAIR paper. And so in repositories, they maintain web addresses in a stable form. Okay, so I think there's another addition about interoperability, about the formatting. So one obvious solution is to use a common file format. For example, like a CSV, a comma separated file, or an Excel file for numerical data. So these files can be written by various programming languages, softwares, and never share data tables as Word or PDF. If you have ever tried to get data out of a Word file or PDF, you understand the pain. And also provide the underlying numerical data for plots and graphs. This, in any case, will be asked during the publication stage. So after the acceptance of a paper, it's often that you're requested to provide the data behind all of the graphs and plots in your paper. Another aspect is convert proprietary binary formats to open ones. This, I think, is a case-by-case basis if you may need this. And so for example, SnapGene is a specific format that is made by the SnapGene software. You would want to convert that to a format that is ingestible by an open database. Okay, reusability. So we all experience this from our exercises, is that it's important to describe the data well. You can write a README file to describe the data, add as many details as possible. So if you have ever gone back to your own data after some time, it may be difficult already to understand your own data and what you have done. So imagine if somebody is completely new to your data. Use descriptive column headers for data tables. So for example, previously in the abbreviation we saw RDL, PHL, we have no idea what those abbreviations are. Maintain tidy data tables, make them analysis friendly. So if you have blank spots in your data table, you can put not a number or some other type of filler to fill it out to make analysis friendly. And metadata formats, some recommendations are here, and we have session on day 3. I think that is, sorry, day 2. That's on metadata. Use commonly known terms and persistent ID and descriptions. Follow the minimum information standards. Okay, so now we get a little bit more into licensing. So for your data, it's important to attach the license files and specify the conditions and the terms in which your data and software can be reused. And what's recommended is the Creative Commons Attribution License, CC BY license. And we will go into that in the next session, a little bit more detail about what the CC BY license is. And there's different flavors of the CC BY license and what is the difference. And for code, a permissive open source license like the MIT BSD or the Apache license 2.0 is often used to make your source code open source. All right, so I think we have another exercise. So let me just stop sharing. Maybe if someone has any questions, please feel free to jump in. Nope. Okay, so trying to find the next side of the, yeah, there we go. I think, yeah. So exercise 2, we will talk a little bit more about an example of FAIR data. So I will copy this into the pad. Okay. So in the pad, we have the next exercise. UniProt is a high quality and freely accessible resource of protein sequence and functional information. So let's have a look at the record of the GFP protein, of course, one of the most famous proteins, and let's look at how the FAIR principles has been met. So does anyone want to comment on, do you see how specifically the FAIR principle has been applied in this case? So I can maybe get started and, oh, sorry, Ashish, checked out, yeah, so I access this file in a different format. So this is in an XML file. So if you do more programming, you may have come across the XML format, is that it has a very standard way of structure of this data. And this is a machine readable way to access this data as well. And so this will make the data very much reusable and interoperable. So I see that you can also download it in different formats, text file, a little bit more difficult, I think, for machine readability, but I think still not too bad. Great. So I think our next exercise is more of a discussion. So I will just copy this. So the FAIR acronym is sometimes accompanied with the following labels. So let's add this. So findable is also referred to as citable, accessible is tractable and countable, and interoperable is intelligible, reusable is linked to reproducible. And so what we can do is to, perhaps in the main room, discuss how these labels directly benefit you as a data creator. Most highly cited articles at EMBL. I think one of them in 2021 was actually just publication of a data set in the form of a preprint of COVID-19 data. So you can see that when you publish data that can be used by a larger community, often it's highly cited, and you can demonstrate the impact of your work in a clear way. Okay, if someone can just look at your data and understand what it does and what it says.
Yes,
Absolutely. Yeah. So Thank you for all of your inputs. So now we are again in a transition period between being fair and the last section, which is about intellectual property, copyright, and licensing. So to bridge between fair and that, we again have a quiz for you. We can take 5 minutes and also take a break at this moment while we're working on this quiz. So we can come back, let's say 257.